Now we're going to look at the situation of independent assortment. And to do that, we have to take a look at two genes that are on different chromosomes. So I'm going to show the chromosomes, one set of homologs as being straight chromosomes, and the other set of homologs as being wavy chromosomes. And what we're going to do is look at the gene that we just visited, the S gene for smooth versus wrinkled seeds, that's on the straight homologs, and we're going to then add in another gene for yellow or green seed, which is the Y gene, and it's going to be on the wavy chromosome. So we're going to start out with two parents. The male is going to be the double homozygous dominant, and the female is going to be the homozygous recessive. They look like this. Male's homozygous dominant, big S, big Y, smooth yellow seeds. The female then is going to be little s, little y, wrinkled green seeds. Alright, so this guy over here is going to have a set of straight chromosomes that have the big S. Here's the other straight chromosome with the big S, and a wavy chromosome that's going to have the big Y, and another wavy chromosome with another big Y. They're going to go through replication. Big Y, big Y. And so now we've got a cell that's ready to go through meiosis. The chromosomes with the big S allele for smooth seeds and the other set of homologs with the big Y allele for yellow seeds. Now, when all is said and done and we take a look at what sort of gametes can be produced, when we just look at what's going on with these homologs, the only possible gamete out of here is big S. And so we know that our gamete is going to have a big S chromosome. And when we take a look at the wavy homologs, the only possible gamete from there is big Y, because that's the only allele that's present. And so we're going to get from the wavy chromosome a big Y. So all of our sperm are going to be big S, big Y. Now when we take a look on the female side, we're going to have, I'll go straight to replication. We're going to have the homologs with the color gene are going to be both small y. Oh, these are supposed to be wavy, aren't they? Excuse me. So those are the homologs with the color genes, and then the homologs with smooth versus wrinkled, straight ones, are going to be little s. Alright, so that's where we start with the cell going into meiosis. And again, if we just take a look at the wavy chromosome, the only possible allele that can go into the gamete from here is little y, because that's all that's there. So we're going to get a wavy chromosome in the gamete that's little y. And in the same way, the only possible gamete coming from the straight homologs is a small s. So all of our eggs are going to be little s, little y, or little y, little s, doesn't matter. So when these combine to form the F1, when we're crossing the male by the female, then the zygote, F1 zygote, is going to have one big S chromosome here, one small s, one big Y, and one little y. So it is going to be, the F1 is going to be big S, big Y, little y, and it's the double heterozygote. Now, the interesting question is, what happens when we self this F1? Well, when we self, remember the gamete formation on the male side is the same result as the gamete formation on the female side, because it's the same plant. So I'm just going to show you what the gametes will be from this plant. So we're starting with big S, big Y, little y. The phenotype of this, by the way, is smooth and yellow, because the capital Y is dominant to the little y, so it's yellow, and the capital S is dominant to the little s, and so it's smooth yellow. The chromosomes going into meiosis look like this. We're going to have one set, I'm going to go right to replication, one set of homologs that are big S, the other straight homolog is going to be little s, and we're going to have a wavy set of sister chromatids that are big Y, and the other homolog is going to have the sister chromatids that's a big Y and with little y. So that is our cell going into meiosis. Now it gets interesting compared to the previous where we had the homozygous parents. Now that we've got a heterozygote, our gametes start to get a little different. If we just take a look at the straight homologs, there are two possible gametes that can come out of this plant. It can either be big S or it can be little s. And there are two types of Y gametes that can come out as well. And so we can have either big Y or little y. Whoop, I'm getting too low to stay in the frame. So big S can also be with little y. These should be wavy. Little s can be with little y. Those are the four types of gametes that can be produced by this cell going through meiosis. Again, it's because we can have two types of gametes with regard to the straight chromosomes, either a big S or a little s, and those are going to independently assort with the results from the Y, which is either big Y or little y. So the big S could be with a little y, or the big S could be with a big Y, or the little s could be with a big Y, or the little s could be with the little y. Those are our four types of gametes. Now, what's going to happen when those combine into zygotes? Again, we do the Punnett square, and we're going to put these gametes across the top for the males and down the side for the females, because remember, we're selfing, and so the gametes will be the same whether it's the male or the female. So let's do that Punnett square. So our gametes for the male were big S, big Y, big S, little s, big Y, little s, little y. And so then on the female side, it's big S, big Y, big S, little s, big Y, little s, little y. And so we're making our little database here. And then interior cells are our zygotes. So here the zygote is big S, big Y, and we can fill in the whole chart this way. Big S, big Y, and so on. And we will get, once we fill this in, a chart that looks like what we see in the lesson. And at the end of the day, when we add up all 16 of these cells, we're going to find a ratio of phenotypes that is 9. Sorry, the camera ran out of battery, so let me pick up where I left off. When you add up all 16 of these database cells, which represent the zygotes from the self-pollination, then you can find out that the phenotypic ratio is 9 to 3 to 3 to 1. What does that mean? It means that there are 9 cells that contain genotypes whose phenotype is smooth and yellow. S dash big Y dash. Smooth, yellow. And there are 3 of those cells that are smooth, green. 3 are wrinkled, yellow, and 1 is wrinkled green. Remember, that dash means that the genotype could be either big Y, or the heterozygote, big Y, little y. Either one, because in either case, the phenotype is yellow. So at the end of the day, you get a 9 to 3 to 3 to 1 phenotype ratio, just like you see in the lesson.